Bueno, buenos días a todos. Es un privilegio tener hoy aquí entre nosotros a José Luis Mascareñas, Premio Rey Jaime I de Investigación Básica de 2025, galardón que recibió ayer. José Luis es catedrático de Química Orgánica en la Universidad Santiago de Compostela desde el año 2005. Allí anteriormente había hecho la tesis, se fue de postdoc a Stanford y después de esa etapa postdoctoral en Stanford volvió primero como profesor titular y luego como catedrático. Ha sido el director del CICUS, un centro que conocemos bien también aquí en la comunidad, desde el 2014 al 2025. Ya este año se ha retirado de la dirección, no sé si se le notará en la sonrisa, la parte de haberse retirado. Y tiene, bueno, numerosos premios. La lista de premios es interminable, los tenía aquí apuntados, pero es que si no, no terminamos la presentación. Entre ellos, una de las cosas que a mí me ha parecido llamativa es que ha dirigido 48 tesis doctorales. O sea, la capacidad formativa de José Luis está fuera de toda duda y es una cosa que de la que pues, yo por lo menos siempre me siento muy orgulloso de las tesis doctorales que se dirige y lo que cuesta y da. Así que 48 es un número nada desdeñable. Y hoy pues nos va a hablar de la química tan diferente que hace, de catálisis dentro de las células. Y nada, José Luis, cuando quieras.
Bueno,
Gracias.
buenos días. Muchísimas gracias, Guillermo. Yo es que me muevo bastante. A ver si me podéis oír. Y, y nada, muchísimas gracias por la presentación tan amable. Tengo que decir que estoy encantadísimo de estar aquí. He estado alguna vez más en Icmol, un centro de referencia para todos nosotros. Quiero agradecer la invitación vuestra. Quiero agradecer, por supuesto, a la Fundación Rey Jaume I por proporcionar esta posibilidad de contaros algo de lo que hacemos en Santiago aquí hoy. Y sí, voy a unirme a esas palabras que estabas diciendo tú sobre lo de la formación. Después voy a cambiar el inglés si no os importa para la charla, pero ahora estos comentarios los hago en español. Porque claro, a mí me acaban de dar el premio de investigación básica. Entonces, yo ya defendía mucho la importancia de la investigación básica y ahora tengo que hacerlo más por la responsabilidad de llevar este premio, ¿no? Y entonces la gente te pregunta mucho cuál es la aplicación, la utilidad de la investigación básica. Podríamos hablar horas de esto, que sería una discusión muy interesante. Digo, una de las mayores utilidades y a corto plazo, es la gente que tú formas. Porque a la larga, el progreso de la sociedad depende de las personas. Depende de las personas. Si lo reflexionáis, depende de las personas. Entonces, si tú proporcionas a la sociedad personas con un alto nivel de formación, estás realmente contribuyendo a ese progreso de una forma completamente útil y completamente aplicada. O sea hay muchas más cosas de la investigación básica dirigida por la curiosidad, dirigida a rellenar lo que yo llamo el bucket del conocimiento, pero una de las aplicaciones inmediatas y a corto plazo son las personas. Y a la larga, si echáis un ojo a por qué los grandes entornos universitarios, académicos e industriales han progresado, es por las personas que tiene. Y por eso tenemos que ser todos defensores, creo, del papel que hacemos cuando formamos a la gente en tesis doctorales y en postdoc. Podría hablar mucho tiempo de esto, pero no es el caso. Vamos a hablar de... Estos días han sido intensos. Estoy un poco sobresaturado. Estos 2 días han sido tremendamente agotadores. Y voy a ver si me acuerdo de lo que tengo que contaros hoy. Espero que sí. Y tampoco sé muy bien cuánto me va a llevar, pero bueno, voy a ajustar para que no sea muy excesivamente largo y contaros un poco todo. Okay, I'm going to switch to English. So let me start by showing you the picture of my center that, as Guillermo was saying, is SICUS. It is a research center that belongs to the University of Santiago, so it's not independent. It has over 200 people working in the areas of synthesis, chemical biology, and molecular materials. So it's very multidisciplinary. And my group is also very multidisciplinary. We work on different topics, but we are synthetic chemists. So we know how to make and how to manipulate molecules. On one hand, over the years, I have been working quite a lot in this field, in the field of transition metal catalysis. What we have been doing here is trying to discover new metal catalyzed transformations with a special emphasis in cycloaddition reactions. But over the years, we have worked also quite a lot in the area of chemical biology and in biosupramolecular recognition. In the last 10, 15 years, most of our work has been in this interface between the two fields. And this will be the main topic of my lecture today, okay? But probably I will also comment to you just only in 4 or 5 slides, some of the things that we are doing in the area of catalysis, because this is also valid as an introduction to the rest of the talk, okay? So let me start the presentation with this picture. Maybe some of you don't know the picture could think, well, this is the map of the subway in Tokyo, but it's not. It's not the map of the subway in Tokyo. This tries to represent a metabolism, a cellular metabolism. What is a metabolism? It's the network of chemical reactions that provide the food and the energy for cells to work. So it's a very complex network. We could say that each of these dots will be a metabolite that is converted to other thanks to a chemical reaction. And as you know very well, these reactions are catalyzed by enzymes, by the catalyst of nature. And in many cases, enzymes work in this way. They work as factory lines. So these are cascade reactions, so that the product of one reaction, of one enzymatic reaction, is the starting material for the next one. This is very typical in metabolism. We have this, for instance, is a typical several steps of glycolysis. Okay. I'm not sure you know, but over 1/3 of these enzymes that we have in our cells contain a metal at their active site. And then they are coined as metalloenzymes. But enzymes and metalloenzymes, of course, are wonderful catalysts. Why they are wonderful catalysts? Well, because they provide impressive rates and turnovers in the catalytic reactions that they catalyze, that they promote. They work with high selectivity, even enantioselectivity, and they work in cells in a very complex medium. And this is a property that very often is not highlighted, it's overlooked. This ability of enzymes to promote their specific transformation in the presence of many other reactions and in the presence of many millions of biomolecules. And this is what we could call orthogonality, or if you want, bioorthogonality. So I want to claim this property of enzyme that is wonderful. But enzymes are not perfect, at least from the perspective of a synthetic organic chemist like me. Why they are not perfect? Well, because they are very good, but to perform the reactions that are of interest for nature, that not necessarily are the reactions that we want to do as humans. Indeed, also the other problem is that enzymes tend to present a narrow scope. So they are not very promiscuous in general. And also, they promote reactions with a limited number of mechanisms. Indeed, if you go to a book of biocatalysis of enzymes and you go to a book of organic chemistry, you will see that the reactions invented by humans are much broader than the reactions invented by nature. Many more reactions, many more types of reactions. So we can be happy that, you know, as humans, even though we don't have so much time like enzymes, we invented many things and many chemical reactions. So this is particularly the case with metalloenzymes, because metalloenzymes use metals like zinc, copper, iron, that are metals of the first row of the periodic table. And these metals work either as redox centers or as Lewis acids. So they are missing opportunities. They are missing the opportunities provided by metals of the second and third row of the periodic table. These metals. rhodium, ruthenium, palladium. And we know that these metals can be very powerful for organic synthesis. These are the metals for typical transition metal catalysis. And we can promote, using these metal complexes, a much broader range of reactions than metal enzymes. And they work also with a very high breadth and generality and scope. And why? Why they are better? Well, because they work through mechanisms that are unknown for nature. Organometallic mechanisms. These are unknown for nature. Nature never cared about organometallic chemistry. So, well, yes, coordination chemistry, but not organometallic chemistry with steps like oxidative addition, reductive elimination, etc. So this is wonderful. But of course, the rates that you get with this type of transition metal catalysis are lower in general, and the turnover is worse than by using enzymes. And of course, we know that to do these reactions, you need to work in organic solvents and under very strict conditions. Okay, so the conclusion is transition metal catalysis is very powerful. And this has been demonstrated by many groups along the last decades. And my group has made a small contribution. I will show only 5 slides with this. We have contributed to this field. We have invented different types of metal catalysis reactions. For instance, we have, as I told you before, we have worked quite a lot in cycle addition reactions like Diels-Alder, you know Diels-Alder, but in this case, metal promoter reactions. And one of the mechanisms that we considered when designing the reaction a long time ago, more than 10 years ago, was this mechanism. We thought, okay, we have systems like this, perhaps we can make a metal promoted CH activation to make a metal cycle. And if you have this metal cycle, you can now add an unsaturated partner and promote the cycle addition, because this cycle addition takes place through typical organometallic reactions, organometallic steps, like in this case, migratory insertion and reductive elimination. You need an oxidant to recover the active catalyst, and then you have the catalytic cycle. So using this mechanistic manifold, we discovered quite a large number of reactions. I will show you the initial ones, that are these ones, in which we were activating this CH bond to make this type of 7-member rings that are quite interesting. These are the intermediate, the Roda cycles, these ones. And we can also make, playing with the ligands, you can go to other type of products. We have been working in this area for many years. At the beginning, Moises was my postdoctoral student in the field. Now he is professor, and we are still collaborating. And for instance, this is just an outline of some of the things that we discovered. We discovered annulation between alkyl anylides and alkynes. This allows to make these interesting indolines in just one step. We discovered enantioselective transformations with palladium. You can make enantioselective cycle addition. This is a fantastic way of making this type of heterocycles, acycles with high levels of enantioselectivity, like this one or this one. Here we found that by using the typical chiral amino acid for the asymmetric induction, if you want to go to a 7-member ring, you have problems, but we discovered a new type of ligands, these ones that present axial instead of central chirality, and in this case the E is very good. Further on, we also discovered a different type of transformations that are slightly different, are not cycle additions, are cyclizations. So this is quite interesting, because if you pay attention to this transformation that is promoted by iridium, in the carbon and the hydrogen of the substrate are in the product. So this means that they are completely atom economical. We don't waste atoms. So, and we are making in the way, we are making a carbon-carbon bond and one cycle. This is the mechanism, the first step of the mechanism, oxidative addition. We can make a great variety of heterocycles. And this is quite interesting that if you use alkenes, this is an alkene, we have 2 double bonds. And we can control which is the double bond that undergoes the transformation. In this case, it's the terminal double bond. You have a small size substituent here to make this product. But if you increase the size and you play with the ligands, then you go to the other double bond, and then you obtain products that contain a chiral stereocenter, a chiral center with high levels of enantioselectivity. And then you can make very interesting heterocycles containing either tertiary or quaternary stereocenters with high levels of enantioselectivity. So this is my last slide in this topic. We are working more than half of my group in collaboration with Dr. Fernando Lopez and Moises Gullias are working in this topic. But I think this gives you an idea of the power of transition metal catalysis to do chemical transformations. So a long time ago, because time goes so fast, I think more than 15 years ago, I started to think about the possibility of translating this power of transition metal catalysis to the same place in which enzymes work, for instance, to living cells. And then the question is why? This was before 2010. So why? Why do you want to do that? Well, the first thing that happens for scientists that work in basic science is curiosity. I just had the curiosity to know if we could really perform a transition metal catalysis reaction within a cell, a living cell. But later on, we realized that this could be very powerful because we are introducing new chemical tools to manipulate the cell behavior. So we decided to approach this challenge. Of course, this is a very difficult challenge because the cell is not like a normal organic chemistry reaction flask. Here we have an organic solvent. Usually we try to avoid water because the water tends to deactivate the metal. So the cell is a very complicated reaction vessel because it's aqueous, salty. Usually transition metals, palladium, ruthenium, they don't like that. Also, there are compartments. Maybe you are not lucky. Your substrates go to the mitochondria, and your catalysts go to the nucleus, and they never meet, so they don't react. Of course, you can have toxicity problems. So there are many problems that you can have. But we thought that the challenge was worthy to approach. And we started to work. And this was, I can tell you, about 2008. 2007, 2008. I started to do some work on that. It was awful because our facilities were very, we didn't have any facility, no microscope, no cell culture lab, nothing. So we had to go to other buildings in biochemistry to do this chemistry. Anyway, we decided to test it. And the first thing that we did is, okay, let's go to the literature to be sure that nobody was doing that before us. And we found this paper by Eric Megers. And this paper was published in 2006. And in this paper, Eric describes a method to remove alloc protecting groups from rhodamine using a ruthenium catalyst. And they claim in the paper that they could do that in the presence of living cells, of HeLa cells. It was not clear at this moment if the reaction was taking place inside or outside the cell. I can tell you that this paper was not cited for 3 years, so nobody was interested in this topic. Indeed, Eric ended up just dropping his work in the field. He's not anymore working in this field because no citation. So this is the problem. So later on, in 2011, Asier Unchiti Broseta, that is here from Granada, I think is from Granada, in the group of Mark Bradley, discovered that they could do the same using palladium nanoparticles, and they could remove this group and obtain fluorescent compounds in cells. Also, it was not clear if the reaction was taking place inside, because these nanoparticles were relatively big, so maybe they had difficulties to go inside the cell. Anyway, these were the precedents, and we started to work in this area as part of other project that was ongoing in my lab on DNA binding. At that time, we were working in the field of DNA binding with this molecule that is DAPI. It's very well known in cell and molecular biology. DAPI binds DNA, like I'm showing here, like the cheese in a sandwich. And the good thing about DAPI is that by binding DNA, it becomes highly fluorescent. So it's very good to stain the nucleus of the cell, because in the nucleus, you have the DNA, and then you can stain the nucleus. So this is the typical staining provided by DAPI. But we knew that for the interaction with DNA, we need this group, imidinions, because it makes hydrogen bonding contacts with the edge of the bases. So what Matteo did, by the way, Matteo is now professor in Cambridge University. This is related with the formation, the training that I told you at the beginning. He's junior professor in Cambridge. So what Matteo did is introducing these red groups here, alox, in the imidinions. And now, if you pay attention, this compound is not in the nucleus. Look at this cell here. So the compound is not in the nucleus, that is this one. It's in the cytoplasm of the cell. So the question for us was clear. Can we do the mega reaction, so the ruthenium catalyzed transformation inside the cell, and then recover DAPI? And we were happy to see that this is possible. If we add the ruthenium, we add a little of thiophenol to help in the deprotection. After 30 minutes, controlling the amounts of ruthenium, the cell was still alive, and we could obtain the natural product. So we were converting an inactive product into an active product. So we were happy with this result. It took us a lot of time to publish this paper because the referees didn't believe it. It was more than 1 year to publish this paper. And we had many difficulties to do experiments because of our facilities. But after that, I got an ERC in this field, and then this was magic because I got money, I got funding, so I could buy my own microscope, I could build my own cell culture in lab, in situs, and it was critical to really progress in the field. So we built everything from zero. So then with this money, I also hired a biologist, José Couceiro, and Maria Tomás as a postdoc, and they were able to do the transformation with other substrates like this one, that is quite nice because this is a very robust substrate, and with this substrate, we could even record a movie. So this is a real-time movie. So these are two sets of HeLa cells, 1 and 2. Now we are going to add our ruthenium catalyst to one of them, and the substrates to both. So this is something that I was showing yesterday on Monday on the colloquium of the Fundación for general public. So I think they understood more or less what I was trying to say. So in these cells, we have the ruthenium and the substrate. In these cells, we have only the substrate. So we should see the product only in the cells that contain the ruthenium. And this is what we saw. So as you can see here, the formation of the green color of the product is only in the cells that contain the artificial ruthenium catalyst. So this is a real-time visualization of a ruthenium-promoted transformation that takes place in a living cell. So this, for us, was a very important development, and from there, we opened a whole program, and we started to demonstrate, we wanted to demonstrate that this chemistry is not only for allelic deprotections, that we can do other types of organometallic transformations. And we indeed discovered a variety of transformations, such as this one that is a ruthenium-promoted isomerization, or this is a formal cycloaddition to give this interesting product, or this is a gold-promoted cycloisomerization. So as you see, in all the cases, we design substrates that are not fluorescent, but the products are fluorescent. And then this is very good because we can monitor the reaction by fluorescent microscopy. So this is the starting material, no fluorescent, and this is the product. In this case, if you pay attention to this picture here, these are cells, but cells treated with this product. And as you can see, maybe you realize from your seat that the product is aggregating outside of the cell because this is a very hydrophobic. It's a self-aggregation dye, and it doesn't want to go inside the cell. It aggregates outside. How can you solve this problem? Doing the reaction inside the cell. So if you add the substrates and you add the ruthenium catalyst, you make your product inside the cell, and then the fluorescence is inside the cell. So this is the idea that products that are not internalized can be artificially constructed, built inside the cells. Maybe this can be useful in the future. Okay. So this is an experiment that we did a few years ago that I like it quite a lot, because in this experiment, we mixed two different substrates, one for the gold catalysis and the other for the ruthenium catalyst. And of course, the probes are not fluorescent, as you can see here. Now, what we are going to do is to add a ruthenium catalyst. And then if we add a ruthenium catalyst, we are going to activate this precursor and to obtain the red color. And this is what we see. And now we add the gold catalyst. And then if we add the gold catalyst, we activate the other product, the gold cyclization. And we have this. And if we add both, we activate both. So what do we have here? What do we have here? Well, we have a cell with the cell metabolism, natural metabolism, but we built an artificial metabolism. Of course, it's very primitive artificial metabolism, so that we have two artificial substrates, two artificial catalysts that are able to work in a concurrent manner inside this living mammalian cell. I like a lot this idea. Indeed, I think this is something that will have a lot of future, is this idea of building artificial metabolism. And we are working on that. We are trying to develop other type of artificial metabolic reactions. I will comment just one with you. So our idea here is mimicking what nature does, the factory lines, so that you can do cascade processes like in the glycolysis. In our case, the idea that Alejandro started, I want to name Alejandro because he was a fantastic PhD student and he went for a postdoc in Sweden and he died suddenly 1 year and a half ago. So we are still shocked with that. And you know, when I remember Alejandro, I feel very emotive because of that. But so Alejandro started, and now it's Alejandra, so we changed the name, Alejandra. And Alejandra is doing this work also, Leonard. So what Alejandra is trying to do is to introduce a probe that is inactive, is not fluorescent. We introduce the probe. Then after undergoing a cascade process inside the cell, for instance, with an artificial metal catalyst and another artificial metal catalyst, or perhaps by integrating a natural enzyme with the artificial catalyst, we generate the active product. So for the generation of the active product, you need the whole cascade, the whole metabolism, the whole artificial metabolism. So this is the idea, and this is just a proof of concept. And so this is the reaction that Alejandra chose. This is inactive. After the ruthenium removal of the allyl groups, we get a phosphate that is also inactive. But after the action of an enzyme, alkaline phosphatase, we get the active product. So Alejandra was able to demonstrate that this works in vitro. So here you have the fluorescence of the substrate. And after adding the ruthenium catalyst and the alkaline phosphatase, you get the product because we observe an increase in the fluorescence. we have all the controls. If you have the ruthenium only, doesn't work. If you have the phosphatase only, doesn't work. And if you have both, but you add an inhibitor of the enzyme, this is an inhibitor of the enzyme, it also doesn't work. So we need the whole process, both catalysts, to do the work. So let's go to cells. What happens with cells? Can we do that? Can we do the tandem process and avoiding hydrolysis, for instance? So this is just the proof of the concept in cells. As you can see, we use these cells here, FG2, because these cells contain a high level of phosphatase. And if you add the ruthenium and the phosphatase, then the system works. You could think, well, maybe you have hydrolysis of some of the initial intermediate, but we have all the controls. So, for instance, you use if you use the inhibitor, then the fluorescence is much lower, but particularly relevant is this slide. Because in this slide, we have 3 different types of cells, FG2, MCF7, and U2S. And these cells contain different levels of this enzyme, of phosphatase. This is something that we proved by assay that is called Western blot. So these contain a high level of the enzyme, this medium level, and this very low level. And you can see, depending on the level of the enzyme, you have more or less of the product. So, well, we extended the reaction to other precursors. It's quite promiscuous. This is to give a product that is also fluorescent, but it's, as you can see, pink. It's much easier to see. And when you send these papers for publication, the easy question is, but this is useful for something. Can you use this system, for instance, to activate a biological property? And we try to demonstrate that this is possible. So in this case, we use this compound. This compound is a known anti-inflammatory compound, but also has been described in dometacin that it cleaves mitochondria. So inside the cell, we have mitochondria, that is the central power of where is the organelle responsible for generating energy in the cell. So if you add this compound to cells, you cleave mitochondria. This is an image of mitochondria. This is one cell. And this is an image of mitochondria that is broken, is fragmented. How do you know that? Because if you compare with the normal mitochondria, you can see it. Here you see the filaments of the mitochondria. But after treatment with this compound, we fragmented it. But this picture here is not really just a normal mitochondria. It's the mitochondria has been treated with this compound that is fully inactive, doesn't do anything. So if you add this compound to the cells, the mitochondria is healthy, no problem. But why this compound? Because we designed it because it contains the bisalyl phosphate that can be cleaved with our tandem reaction, and this is a self-immolative group. And after the self-immolation, you get the active product. So the idea here is you add this compound that is inactive, and after the tandem, you get the product that is active. And this is what we tested, and this happens. So when you have this substrate, nothing happens, but after the tandem reaction, you observe the fragmentation of the mitochondria. Of course, if you use cells that do not contain the phosphatase, it doesn't work. So this is a cell-selective process. We can do this process even in a more selective manner. We designed this system here. I explain you. So this system contains 3 functionalities. This is the ruthenium catalyst. This is a fluorophore. And this here is called a RGD. It's a tripeptide that is well known to target integrins that are proteins in the membrane of cells. So by targeting integrins, if the cell contains a high level of integrins, the compound goes inside the cell. If the cell contains a low level of integrins, it doesn't want to get internalized. So what we found, this is what happens. If you have these cells, MCF7, with a low level of integrins, the compound doesn't go inside. But if you have this other cell, HeLa, with a high level of integrins, the compound goes inside. And we can see that by fluorescence, because we have a fluorophore here. So in these cells, we have ruthenium inside the cell. In these cells, we don't have ruthenium inside the cell. So we tested the reaction, and we see that only in HeLa cells we can observe the transformation, but not in the other cells. So this is an extremely cell-selective formation of an active product, and this is the conclusion. So this is like a logic gate, an AND logic gate, so that only when the cell contains integrins and the phosphatase, we have activity. However, in other cells that fail with one of these components, we don't have activity. This can be quite interesting from the point of view of biomedicine, because this will allow to activate, for instance, an anti-cancer compound only in cells that contain both inputs. Okay, so let's go to this, go back to the metal-promoted transformations. So as I told you, many people thought at the beginning that it would be impossible to promote transition metal catalytic reactions inside living cells. We demonstrated that it's possible. Of course, the efficiency is low. The catalysts, and particularly palladium catalysts, are readily deactivated. Why? Well, it makes sense, because for the organometallic chemistry, you need vacant sites in the metal. If you don't have these vacant sites, then the reaction doesn't occur. And when you have these vacant sites, you have a problem. It's natural metabolites like glutathione can occupy these sites, and then the catalyst became deactivated. So what people are trying to do, now when Megas was working, nobody was working in this field in 2006, 2007. Now there are many groups working in this field. Still, the community is small, but there are many groups, particularly in Asia, in China, and in Japan. So what are these people trying to do? Well, trying to protect the metal with protein scaffolds and make metalloenzymes and see whether with these metalloenzymes they can do these transformations. But metalloenzymes that work inside the cell. I will go briefly to our effort in this area, and we are trying to make pallado-metalloenzymes, so complexes with palladium, because a few years ago, we had discovered that some peptides and proteins that contain histidines in this position 1, 4, are able to make this type of complexes with palladium. These are typical palladium 2 complexes. And what we have discovered here is something quite fascinating, is that when you don't have the palladium, the peptide doesn't want to go inside the cells. However, when you have the palladium, when you have the palladium, the compound goes inside the cell, so it's internalized. So the first time that I saw this is a palladium-promoted internalization. This is very strange. I told my student, Soraya, that is from Valencia, by the way. Soraya, are you sure about that? Is the palladium promoted cellular internalization? And she tested many times, and it works, always works. So what we think is happening here is that the palladium is facilitating the formation of the alpha helix, and then when you have this compact structure, it goes easily inside the cell. So palladium promoted internalization, but also peptide promoted internalization of palladium. So we have palladium in the cell, that is somewhat protected by the peptide, by the mini protein. And then we thought, well, maybe this works in catalysis. Let's try. And we tested that in this reaction, that is a depropagylation. And this is the first result. It didn't work. We didn't observe the formation of the product because no fluorescence. And then we thought, okay, it doesn't work. The palladium is not doing his role. But we realized that this palladium is bound to a bipyridine. So there are no coordination sites here. We did vacant sites in this position where you want organometallic activity. And then we found indeed that if you use other palladium that contains a COD ligand, a cyclooctadiene ligand, the reaction works very well. And importantly, if you use just the small molecule, the palladium dichloride, it doesn't work. So the peptide is favoring the reactivity by protecting the palladium. So this result led us also to initiate some program in this area. I have 2 persons working in this area in collaboration with my colleague Eugenio, trying to make other metallopeptides or other mini protein palladium complexes that can really work better than this one. And just briefly, I can tell you about one of them that we have already published, that is using this type of proteins, mini proteins, because they are very well folded, as you can see here. And we thought that we could take advantage of this folding to introduce our histidines for palladium coordination. And this is what we did. And we were able to even to characterize our compound. So in collaboration with Maria and Jaderia Marichal, we were able to demonstrate that we can make these pallado mini proteins. And the good thing is that these pallado mini proteins are also very well internalized into cells, as you can see here. Look at the color. Not so good like the other one, but still we have the compound inside the cell. So this is a rhodamine 404, so we can monitor the internalization. So this type of system with the palladium is inside the cell. We even had this movie that is quite nice, where you can see the cell that has been stained. The nucleus has been stained with DAPI, and these red spots that you can see are vesicles with our palladium mini protein. This is a movie here where you can see the cell, each cell with the cytoplasm, in which you have the palladium mini protein in this type of vesicles. So the question is this palladium mini protein complex still active? And yes, it's active. It works very well. So when we have it in cells, we observe activity. We were able even to isolate by using mass spectrometry and HPLC. We were able to isolate this product and quantify the product. And we found that we have turnovers about 10–15%. That is not so bad for a pallado enzyme working inside the cell. We are still working in this area, and we hope that in the near future we can really make artificial metalloenzymes that work efficiently inside mammalian cells. Also, and this will be the last part of the last 10 minutes or so. I will show you other part. So let's go back to this picture. I told you before that this is the problem with the metal catalyst, that you have vacant sites, and these vacant sites can coordinate natural metabolites, and then you can deactivate the metal. So now other people are working, ourselves also are working trying to solve this problem. But when we saw this, we also considered the possibility of using fully saturated metals. So metals that have no vacant sites, like these ones. But of course, if you have these metal complexes, you cannot do organometallic chemistry. But what can you do with these metal complexes? I'm sure that many of you are thinking about the same. These are typical metal complexes used for photocatalysis. And we were excited about the possibility of using photocatalysis in living systems. Why? Well, because we could develop new type of transformations. Also because we know that this type of complexes can promote photocatalytic processes in vitro in organic chemistry using visible light. And we thought that visible light is good for living systems, particularly near infrared light, but green light or red light is also okay. And these catalysts present long-lived excited states, and this is also quite good if you want to use these compounds for photocatalytic chemistry in living contexts. Are there precedents on this? Yes, there are some precedents. So this is something that perhaps some of you know, photodynamic therapy, that is the use of photocatalysts to generate active oxygenous species, and this has been used quite often to damage cells. This is a therapeutic tactic even has been used in the clinic, particularly, for instance, in eye diseases, in macular degeneration. You can use this type of photodynamic therapy. There are a few examples, very few, in which photocatalysis has been used in living context to remove protecting groups. They will be the protecting group, and you get the active compound, but only a few examples by Winsinger. And recently, there is a new research field that has been inaugurated by Macmillan. Macmillan again, you know, Macmillan was, as you know, one of the main players in the field of photocatalysis, but also is now working at this frontier with cell biology. So with Macmillan, in collaboration with Merck, a few years ago in 2020, demonstrated that if you install a photocatalyst in the surface of a cell, then with light and a reagent that is a carbene precursor, you can generate an active carbene nearby, near the photocatalyst, and with this active carbene, you can label proteins that are close to the photocatalyst. So this is a very nice tactic for micromapping of the proteome that is near the photocatalyst. So these examples, in some way, were showing that you can use photocatalysts in living contexts, but nobody had used photocatalysis for synthesis, for making bonds in living contexts. And then we thought, okay, this could be interesting. You know, can really we use typical photocatalytic transformation of organic chemistry, translate them also to living contexts, to living cells? So this was the idea, and we knew that for doing that, we had to choose very well the substrates and the reaction that you use. And we considered this one because it's an intramolecular reaction, so it's not so difficult, because you are making an indole that is an interesting product from the biological point of view, and you're using this photocatalyst here, ruthenium-3-BP, that is very typical in photocatalysis. And this is the mechanism proposed for the reaction. So the mechanism involves the generation of a nitrine because of an energy transfer process. So you excite the photocatalyst, this photocatalyst excites the substrate, you release nitrogen, you form the nitrine intermediate, and then there is a CH insertion here to give the product. So we thought, can we perform this transformation in a cell, in a living cell? So the first thing that Cynthia did was demonstrate that the reaction is compatible with water and is compatible with lysates, with cell lysates, as you can see here, and even with whole HeLa cells. And the more important thing is that we developed a protocol for performing the reactions in cell and analyzing the result by HPLC-MS. And we observed in the first experiment, we observed the formation of the product. But the important thing is that as you increase the concentration of the photocatalyst, you observe more product. So this is demonstrating that we do have a photocatalytic transformation inside the cell. Even Cynthia was able to isolate the ruthenium tris-BP catalyst that we have inside the cell. So if you pay attention to these two columns here, you can see with this amount of ruthenium tris-BP inside the cell, you have this amount of product. So it means that we have turnover. We have a catalytic process, a photocatalytic process. Of course, as always, we like to have something that we can monitor by fluorescent microscopy, and Cynthia demonstrated that this is possible by using this substrate that is more conjugate, and then we can make a product that is blue. The problem that we found is that when we went to do the experiments, in this case, with blue light, the reaction does not need the catalyst. It also works without the photocatalyst. However, by using low energy light, like green light, you can get the reaction. So we went to the cells and we found that the system works again very well. You can see that by increasing the amount of the photocatalyst, in this case eosin Y, you observe more product inside the cell. So we have, again, a demonstration of a green green light promoted photocatalytic process inside the cell. We went further, and recently we made this other catalyst in which the eosin is linked to a RGD moiety, and this photocatalyst goes much better inside the cell than just free eosin Y. And with this photocatalyst, we were able to obtain quite efficient transformations inside HeLa cells. For instance, if you compare this photocatalyst, eosin RGD, with eosin, this is much more efficient. And also is cell selective. So you have other cells like MCL7 that lack integrins, you have no photocatalytic transformation. So we can say that this is the first demonstration of a bond-forming synthetic process taking place inside the cell, promoted by a photocatalytic reaction. And we hope that this can be further exploited in the future. And so I have only 5 minutes. In 5 minutes, I finished. So let me comment to you that we were surprised to find that when we did this reaction, that is the same as this one, but you can see that in this case we use a cis derivative instead of trans. If the reaction works through an insertion process, NH insertion process, this product should not work. However, it works. So the mechanism is wrong. So we thought, this doesn't make sense. But Cynthia repeated this experiment many times. So this is not really a nitrine insertion. It has to be a different mechanism, because the yield is very high in this case, 62%. And it's not possible geometrically, the insertion here is not possible. So we did some calculations, Marta did some calculations, and we found that indeed this is not the mechanism. Well, we are not 100% sure, but the mechanism does involve the formation of the nitrine. This is the nitrine, the triplet nitrine. She investigated the pathway to the product, and this pathway is very difficult because of the high energies. This is through the triplet to the triplet nitrine. However, what she found is that this is the very easy pathway to the product, this one. So this triplet nitrine that we have here goes through an intersystem crossing to a singlet, and this is energetically quite accessible. And when you have this singlet here, what happens is a radical addition to the double bond, radical addition to the double bond. And this radical addition gives to the product. a radical addition. And then this word, radical, switched on our lights. Radical chemistry in cells. Why not? Can we do radical reactions, artificial radical reactions in cells? Can we handle radicals in cells to make synthetic reactions? Well, this is something that sounds exciting. And then I have several people working in the group right now in this idea. And I can comment just to finish. I promise to finish 2 cases. This is a radical cyclization that is also promoted by a photocatalyst, but it's not an energy transfer process. It's a photoredox process. There is a single electron transfer here. So single electron transfer, you generate this immunyl radical, and there goes the bond-forming cyclization. This goes to the product. And here you regenerate the electron that goes back to the photocatalyst. So it's redox neutral, but it's a radical addition of this radical to the aryl ring. It's a nitrogen-carbon bond-forming reaction. And this is, we found that this works in vitro in presence of water. And most importantly, we found that this works also in living cells. The yields are not very high. We found yields about between 5 and 10% inside the cell. That is not bad, but it's a radical process. And you need everything. You need the photocatalyst, the light, because if not, you are not generating the radical. If you have all the controls here, the reaction doesn't work. So we are quite excited with this because in some way we are generating a new area that is an area that we could call bi-orthogonal synthetic radical chemistry. And as I told you, we have several examples already ongoing, and this is also very exciting because this one, in this other process, we are generating a different radical, aryl radical. So the idea here is that you have a single electron transfer process, you promote the release of the leaving group to generate an aryl radical that can be, for instance, reducing by a typical HAT process. And this is quite interesting because if you think that this is a prodrug, you could be converting a prodrug into a drug using a radical process. And this type of conversions of prodrugs into drugs that I showed you, for instance, in the case of indomethacin, typically require the presence of polar groups. But with this system, we could use a carbon-hydrogen bond for this idea, for this concept of prodrug activations. So is this possible? Can we create a new type of a strategy for the activation of drugs based on the unmasking of aromatic CH bonds instead of polar groups, typical polar groups? And Mauro and Adrian are demonstrating that this is possible by using this type of precursors in which we have a tyrant training group because we can introduce the group. This is like a protecting group. So you are deactivating the system, and then with a photoredox process, you can go back to the CH bond. We apply this for different types of precursors, and we are investigating also this in vivo in cells, and we demonstrated that this is possible in cells with this compound, for instance, that is called boscalid. Boscalid, we can deactivate, this is completely inactive, and we can make the natural product, boscalid, inside cells, as you can see here. Unfortunately, boscalid probably was not a good choice because boscalid is a cytotoxic compound, but it's not very cytotoxic. So we didn't observe any biological change. However, what we found is that boscalid is a very potent antifungal agent. And then we thought, okay, can we switch on the antifungal activity of boscalid? And this is the last result, the last slide. You can see this is completely inactive. You can see here in this agar plate that the fungus is growing perfectly. And now with green light and eosin Y, NADH as reductive agent, we can generate boscalid. As you can see now, the fungus cannot grow. So this is an in situ activation of an antifungal agent using a photoredox catalysis that involves radical chemistry. So this is the summary. I hope I have been able to convince you that, in contrast, many people thought it is possible to make transition metal catalysis in cells, in living systems. Of course, the field is still in a very early stage. We need to improve the efficiency of these systems. I'm convinced that in 10 years, there will be metal enzymes working inside cells. But metal enzymes with ruthenium, with palladium, with gold, etc. Many people are making artificial metal enzymes, but not to work inside the cells. Okay. So also, I showed you some artificial metabolisms, and hopefully this can be useful in the future for a field that is called synthetic biology. And also, I showed you the first step of a new field that we could call bioorthogonal photocatalysis of radical synthetic, in cell radical synthetic chemistry, that we hope that can be further developed in the near future. Indeed, in this area, I have 4 persons working in this area right now, and hopefully in the future there will be more people working on that. And this is a picture of my group. I think this picture is already more than 1 year old. I want to highlight mainly the role of Maria Tomás, because at the beginning she was postdoc in my group, and now is last year Ramón y Cajal, and she is co-leading with me this topic, this area. Moises is the one leading the area of CH activation chemistry in transition metal catalysis. So again, a picture of the Siccus, and this is the end. I want to thank you for your attention. I talk about many things, I'm sorry. But I will be very happy to answer any doubt or question, or to have any debate that you want. Okay, thank you.